Good afternoon, everyone. Today's topic will be nervous tissue, histology of nervous tissue. Okay. So, what exactly is the nervous tissue and what you have to study? That we'll discuss today. Now, as I have seen from nervous tissue, very less question used to come from nervous tissue, especially for histology. Okay. Now, there is a region that is neuroanatomy. Okay, so once the head and neck portion is over, at that time we'll start with the neuroanatomy. So when we'll be discussing about the neuroanatomy, at that time directly the parts of the brain, parts of the spinal cord, all those things will be started. Okay, now the main problem is the important points which we'll be discussing now, the nervous tissue. Okay, same thing will not be repeated over there in nervous system or the neuroanatomy part. When it will be started, at that time directly you will start from either spinal cord or from brain. Most of the time from spinal cord it is started. Okay, so there are some terminologies which are associated with the nervous system, and most of the things we'll discuss today. Okay, but the nervous system part, that will be discussed at last phase. Okay, when just before the final exam, at that time all those things will be discussed, both theoretically and practically. So the nervous tissue is a specialized tissue which shows two important properties. What are those properties? That is the irritability and conductivity. That means it is giving response to the stimulus, and along with that it can conduct the impulses. Okay, the stimulus which will give, after that the impulse is generated, and that impulse can be transferred from one tissue to another or from one part to another by the nervous tissue. So these are two important features of the nervous tissue: irritability and conductivity. Okay, now because of these properties it can receive information about the external and environment, and it can transmit those information from one place to another. That is the speciality of the nervous tissue. Okay, now can you give an example? Suppose you are touching any hot object. Suppose a object is placed over here, it is very hot or it is very cold. Now if it is very hot, if you are touching the object, you will remove your hand immediately. Okay, why? Because from this hot object, the impulse which are getting by the nerve fibers, it is being carried to the nervous system. It is being carried to the central nervous system. Okay, so the information from this part is going, that means it is conducting, and this hot object, it is acting as a stimulus which is giving the stimulus, and that stimulus with, in the form of an information or impulse, it is carried. Okay, so that is the example, or you can say main function of the nervous tissue. Next, nervous tissue has the property of the ability to conduct impulse rapidly from one part of the body to the another. Now, if you see in our brain, a total number of neurons are present, approximately 1 trillion neurons are present in our brain, approximately. Okay, and the nervous tissue which is there, it is mainly composed of two types of cells. These are the neurons and the neuroglia. Okay, so from the beginning I am telling you that whenever the word tissue will come, please note that cells will be present. Okay, cells will be there. Now here also you can see this is nervous tissue. So in this nervous tissue mainly two types of cells are present. What are these two types of cells? Neurons and neuroglia. Okay, so what are neurons? Already you know that these are the specialized cells which are the structural and functional unit of nervous system. Neurons, you have studied a few things about the neurons in 10-12 standard. So neurons are the structural and functional unit of nervous system. Today we'll see the structure of neuron also. And what are the neuroglia? These are the supporting cells, supporting connective tissue cells which are present around the neurons. Okay, those are the neuroglial cells. Okay, so these are the two important cells which are present in the nervous tissue. Next, can anyone tell me the classification of nervous system? Do you know the classification of nervous system? So here you can see the nervous system classification: central nervous system, peripheral nervous system, and autonomic nervous system. Okay. In central nervous system, we are having brain and spinal cord. Peripheral nervous system, we are having peripheral nerves and ganglia. And in case of autonomic nervous system, sympathetic and parasympathetic. Okay. Now here, these are the different parts of the brain. Okay, all these are the different parts of the brain. Next, in the peripheral nervous system, in peripheral nerves, two types of nerves are there: cranial nerves and spinal nerves. Okay, and in autonomic, we are having sympathetic and parasympathetic. Okay. Now, have you noticed one thing? This particular terminology, ganglia. Anyone, have you heard about this ganglia? Where you have studied about ganglia? No. Till now, you have not studied this in physiology. That means you have not studied till now. Okay. Today we will see what are those ganglia. Very important. So this is the overall classification of nervous system. Now, in the nervous system, in the nervous tissue, what I have said, how many types of cells are there? Two types of cells are there: neurons and neuroglial cells. Okay. So first we will see about the neurons. So if you see the cellular organization of the nervous system, okay, highly complex nervous system consists of only two types of cells, that is neurons and neuroglia. Okay, so you know that the nervous system is highly complex system. Only two types of cells are there. So neurons are the structural and functional unit. And if you see the function, they are excitable cells which are specialized for reception of stimuli and conduction of the nerve impulse. This is the main function of the neurons which are present in your body. Different types of neurons are there. But ultimately, their goal is, what is the goal? Reception of the stimuli and conduction of the nerve impulses. Next is the neuroglial cells, also called as glial cells. These are the supportive cells that support the neurons both structurally and functionally. Okay, these are the supportive connective tissue cells present in the nervous system. Those are the neuroglia or glial cells. So there is a list of glial cells. Along with that, the important functions and structure that also you have to study. Okay. See, repeatedly I am telling you guys, once we are discussing this, next these things will be applied, or you have to apply these things when we will study the neuroanatomy. At that time, this particular part will not be discussed. Now, if you see the neuroglia, okay, now there is a very interesting concept about the neuroglia. If you see the neuroglia, these are 5 times more as compared to the neurons and account of more than half of the weight of the brain. इसका मतलब क्या है? The number of neuroglial cells are more as compared to the neurons which are present in the central nervous system or in the nervous system, not in the central nervous system. Another important point, do you know that the neurons which are present in our body, they cannot multiply? I hope you know that the neurons which are there in our body, they cannot multiply. So mitosis nahi hota hai. Okay. Now the question is, then how come a person is getting brain tumor? No. But how? No, I am talking about without metastasis. Purely, suppose different types of brain tumors are there. Okay, among them, if a cell is not dividing, then how we are getting brain tumors? Think. Deposition? No. In tumor, cell doesn't deposit. Yes, very good. So sometimes you have to use your common sense also. As I have mentioned that nerve cells or the neurons do not multiply, but the neuroglial cells can multiply like a normal cells of our body. Okay? So when it becomes uncontrolled, at that time different types of tumor occurs. Okay, I will give you a common example: schwannoma, which is a tumor of Schwann cells. Okay, glioma is there, blastoma is there, like this different types of tumors are there. Okay, so those are occurring from the neuroglial cells or the glial cells, not from the neurons. Okay, so Now we have to discuss about the neuron structure of the neurons. So neurons are the structural and functional unit of nervous system. Okay, so you know the main function, that is the reception, integration, interpretation, and outward transmission of the information. These are the main functions of the neurons. Okay, reception, first it will receive, then it will integrate, then it will interpret, and then it will transmit the information. Okay. Next, they conduct nerve impulses over long distance at a great speed. And if you see the nervous system, vast number of neurons are present, approximately 10 to the power 12 number of neurons approximately in our nervous system. Okay. Now, if you see the neuron, okay, mainly it is made up of three parts. Now, what are those three parts? Nerve cell body is there, dendrite and axon. I hope you have studied all those things in 10, 12 standard, right? Now, so can you identify the diagram? Yes. So this is a ideal diagram showing the structure of a neuron. Okay. Now the dendrite and the axon form the processes of the neuron. Please keep that in mind. The dendrite and the axons which are present, they are forming the processes of any neuron. Now dendrites are forming the short process, axons are forming the long process. Okay. Next, dendrites and axons are usually called as nerve fiber. Now, have you heard about this terminology, nerve fiber? I hope you have heard about nerve. जैसे muscle fiber होता है, वैसे nerve fiber भी होता है. Now, if anyone asks you what is the difference between neuron and a nerve fiber, what will be your answer? Neuron and nerve fiber, difference क्या है? No, for that you have to understand. जैसे न्यूरॉन का डायग्राम मैंने यहाँ पे प्रोजेक्ट किया है. You can see the diagram of the neuron and you can see the parts of the neuron also. ठीक है? And we'll be discussing about the structure of the neuron. Now the question is, which is the nerve fiber? Which part is called as nerve fiber, or what is the nerve fiber? तो see, जो न्यूरॉन होता है उसका एक शॉर्ट प्रोसेस होता है, एक लॉन्ग प्रोसेस होता है. लॉन्ग प्रोसेस इज द एक्सॉन, शॉर्ट प्रोसेस आर द डेंड्राइट्स. ठीक है. Now together this short process and long process together they are called as nerve fiber. That means this axon and the dendrites together, these are called as nerve fiber. Okay. Next, so what you have to know about the structure of the neuron, or what you know till now, what you have learned about the structure of the neuron. अभी तक क्या पता है? If you can recall what you have studied in class 12 standard. Yes? No, first you have to see, in case of neuron, the most important part is the cell body. ठीक है? So here you can see this is the cell body, which is irregular shaped. Okay, cell body is present which is irregular shaped. Now if you see the cell body, like any other cell, plasma membrane is there, and inside that cellular granules are present. Okay, you can see over there. Now the cytoplasm consists of large number of nissel bodies, neurofibrils, mitochondria, Golgi apparatus. Now among them, if you see, the nissel bodies and neurofibrils are found only in the nerve cell, not in any other cell of the body. That makes it different. These are the two things which are present in the cytoplasm of the cell body of neuron. Nissel bodies are also called as nissel granules and neurofibrils. Okay, बाकी किसी सेल में भी नहीं रहता है. So that is the speciality of the cell body of the neurons. Okay. Next, if you see the nucleus, it is having only one nucleus which is centrally placed. Okay, nucleus has one or two prominent nucleoli, but doesn't contain centrosome. So in the beginning I have told you the cells, the neurons, it cannot multiply. Okay, why it cannot multiply? As you can see, nucleus does not contain centrosome. That's why it cannot multiply like other cells of our body. Okay, so these are the point. I know you have studied, you cannot recall it right now, but all those things are there in the 10-12 standard. You can check. Now, if I ask you what is neurofibrils and what are Nissl bodies, then I don't think so you can answer. Okay. So what are these neurofibrils? As you can see, these are the neurofibrils or Nissl granules. Okay. So these neurofibrils are thread-like structures present, okay, in the cytoplasm. And if you see the appearance of the neurofibrils, they are network-like. Okay, they are having network in the soma of the nerve process. Now, the presence of neurofibrils are the important characteristic features for the neurons. And neurofibrils consist of microfilaments and microtubules. Okay, ये जो neurofibrils दिखाई दे रहा है, you can see the neurofibrils. These neurofibrils are made up of microfilaments and microtubules. Now, the main function is to provide the structural support and axonal transport. Structural support to the cell body and axonal transport, जो impulse transport होता है through the axon. In that, it is helping the neurofibrils. Okay. Next, after the neurofibrils, there is an important structure that is called as Nissl granules, or also called as Nissl bodies. Okay. Now, these are basophilic granules present in the cytoplasm. When you will take the cell, if you do the H&E staining, after that you will see the basophilic granules. Okay. Now, those basophilic granules are present in the cytoplasm, as well as it is also present in the dendrites also. But these basophilic granules, they are absent in the axon hillock and in the axon. Okay, these are the Nissl granules. Now, what is the main function of the Nissl granule? Synthesis of proteins, different types of proteins. Cell ka protein synthesis is very important. You have studied earlier and you will be studying in biochemistry. So, this protein synthesis is, in case of these nerve cells, these are done by the Nissl bodies. Okay, so protein forms in the soma are transported to the axon by axon flow also. And after that, once it is formed, then it has to be transferred. So through axon hillock and through axon, it is transferred to the other cells also. Okay, so that is the main function of Nissl bodies. Next, as usual, we are getting mitochondrial Golgi apparatus. Okay, and if you see the mitochondrial Golgi apparatus, they are having same function as the normal cells. Okay, the functions of mitochondrial Golgi apparatus, normal cells में जो function है, same functions are there in case of neurons. Okay. Next is the dendrites. So you can you see the dendrites? ये देखो, these are the dendrites. Okay. So these are highly branched, short tapering processes. Okay, short branched, highly branched tapering process. Now these dendrites either end as a sensory receptor in a primary sensory neuron, or is forming synapses with the neighboring neurons to receive the stimulus. This is the you can say termination of dendrites. Okay. So the dendrites which are present, first of all, they are highly branched, very short tapering process. Okay, and the termination is maybe as a sensory receptor, or it may synapse with the other cell, neighboring cell, to get the stimulus. Now, in some neurons, the smaller process of the dendrites bear numerous minute projections, and these are called as dendritic spines. Not in all cases. Now, in case of dendrite, okay. In physiology, you have not studied the synapse chapter. Okay, you will be studying. Now, when you will study the synapse chapter, at that time you will come to know that different types of synapses are taking place. Okay, 4 different types of synapses are there. Now, when the synapses are taking place with the help of the dendrites, at that time when the conduction is taking place, the impulse is passed from the dendrites, it will go towards the cell body. Okay. So here you can see the dendrites conduct the nerve impulse towards the cell body. That is called law of forward conduction. This we will study in physiology. Okay, physiological topic. And in case of axon, it is away from the cell body. ये difference है axon and dendrite का. Okay, that means these are the dendrites. Okay, now imagine another neuron is present over here, and with the help of the dendrite, the synapse has taken place. At that time, what will happen? The impulse will go towards the cell body. Okay. In case of axon, if the axon is making the synaptic contact, then what will happen? The impulse will go away from the cell body. Okay. That is the difference. Important difference. Next is the axon. So this particular part is axon, as you can see. Okay. So axon is a cone-shaped portion of the cell, which is a continuation of the axon hillock. So here is the axon hillock, and then cone-shaped continuation of the axon hillock, which is cylindrical in nature. Now the terminal part is having small branches, that is called as telodendria. You can see the terminal part, small branches are arising from the terminal part. These are called as telodendria in case of axon. Okay. Next, in the terminal part, presynaptic terminals are there, or terminal buttons are there, which you can see over here. Okay. So terminal branches are called as telodendria, and terminal parts, these buttons or swellings are present. That is called as presynaptic terminals, which helps in the formation of synapse. That is the part of axon. Okay. Next, the plasma membrane bounding the axon is called exolemma, and the cytoplasm which is present inside the axon, that is called as exoplasm. Okay. Now, in case of exoplasm, cell organelles, everything is present except the ribosomes. Now, the protein synthesis which are taking place, those are transported through the axon terminals by the exoplasmic transport. And here the impulse is traveling away from the cell body. So that is one of the important difference between the axon and the dendrites. Now, these are some important aspects of the axon. For example, axon constitute the nerve fiber, as I mentioned earlier. Okay, so you have to study about the tracts in case of spinal cord. When you study the spinal cord, at that time you'll come to know about the tracts, ascending tracts and descending tracts. Those are nothing but a collection of the nerve fibers. Okay, various information, or you can say impulses, are carried, okay, towards the brain, and from brain it is going towards the periphery or into the different organs with the help of the tracts. And these tracts are nothing but the collection of nerve fibers. And in case of peripheral nervous system, those nerve fibers are also called as peripheral nerves. Okay. Now, an axon can terminate in two ways. जो axon होता है, उसका जो termination होता है, there are two ways by which the axon can terminate. First, if it is in central nervous system, then it terminates by coming in intimate relationship with another neuron, which is forming the synapse. So one neuron, another neuron, they are synapsing, and thus the axon is terminating, and that is taking place inside the central nervous system. Now, if it is outside the central nervous system, now axon may direct end in an effector organ. For example, the nerve can go and supply the muscle, or it can go and supply the gland, or it can go and supply any of the organ. In that case, it is outside the central nervous system, and it is going and supplying the organ, effector organ directly, where it is terminating. Okay. So here you can see outside the central nervous system, or it may end by synapsing with the neurons which are present in the peripheral ganglion. So this is the two ways by which an axon can terminate. Okay, and all those things, as I am repeating, will be repeated again, or will be used, or will be utilized again in the nervous system chapter. Next, here is a difference between the axons and the dendrites. So I'll request everyone to go through it so that you can understand the difference between the axons and dendrites, whatever we have discussed. Okay, once you go through it, you will understand easily the axon or dendrite में difference क्या है. There will be no difficulties. I hope it is visible. Last bench. ठीक है, तो anyone can you read what is written? हाँ बोलो. That means it is long, a thin process of the nerve cell. Okay, and if you see the dendrites, these are multiple short, thick, and tapering processes. Okay, which terminate near the cell body. So it is terminating away from the cell body. It is terminating near the cell body. Okay, you have seen the diagram also. Next, this rarely branch at right angle. So you will not see the axon is branching at a right angle. Okay, in some cases axon collaterals are there, but uska bhi branching perpendicular nahi hota hai. Here you can see dendrites are highly branched. Their branching patterns form a dendritic tree. So branching is highly branched. That's why it forms a dendritic tree. Then it is having uniform diameter and smooth surface, and the thickness of dendritic tree reduces as it divides repeatedly. So thickness here it is uniform, here it is not non-uniform. Okay. Now here nissil granules are absent, as I mentioned earlier, but here nissil granules are present. Now the impulse, how it is carried. In case of axon, the nerve impulse is traveling away from the cell body. Here it is traveling towards the cell body. And this is in the case of synapse, when the synapse are formed. Okay, so for that you have to understand the synapse concept of the synapse and the types of synapses. Okay, and that is a purely physiological topic that will be discussed in physiology. Okay. Now, I hope you have heard about myelin sheath, right? Now, can anyone tell me what is, where, can you show me where is the myelin sheath over here? That yellow color part is the myelin sheath. But actually, what is the color of myelin sheath? Can you tell me? White color. Very good. And because of this myelin sheath, only the nerve fiber appears white in nature. Okay, myelin sheath रहता है, इसलिए जो nerve fiber है, इसका appearance white दिखता है, completely. ओके यहाँ पे येलो दिया है सो दैट टू मेक यू अंडरस्टैंड कि हाँ माइलिन शीथ रहता है. ओके डोंट थिंक दैट इट इज येलो इन कलर. सो what is myelin sheath? So it is an insulating sheath surrounding the axons of the myelinated cells. That means myelinated axons. Now myelin sheath is produced by oligodendrocyte. Anyone have you heard about this oligodendrocyte? No, don't worry. Now two things are there which you have to follow. Now if the nerve fibers are inside the central nervous system, okay, then it is the myelin sheath is produced by oligodendrocyte. If it is outside the central nervous system, then it is formed by the Schwann cell. I hope you have heard about Schwann cell. तो जो पेरिफेरल नर्व है, पेरिफेरल नर्व में जो माइलिन शीथ फॉर्म होता है, that is formed by Schwann cell. Same thing when it is inside the central nervous system, then it is formed by the oligodendrocyte. Oligodendrocyte is nothing but a glial cells, neuroglial cells or supporting cells. Okay. So these are the areas from which you can expect short questions. ठीक है, यहाँ से I am talking about MCQ. Short question मतलब MCQ. Next, this myelin sheath is a thick lipoprotein sheath that insulates the myelinated nerve fiber. Okay, now why the insulation is required? Because whenever the nerve impulse is passing or whenever the nerve impulse is flowing, at that time myelination is required, or else the conductivity is not smooth. ठीक है. Next, myelin sheath is non-continuous sheath. Regular intervals are present, and those are called as nodes of Reinbeer. Already you know that. Continuous नहीं रहता है, बीच में gap रहता है, and those are called as nodes of Reinbeer. Now the area where the myelin sheath is absent is called as nodes of Reinbeer, and the segment between two nodes is called as internode, as you can see in the diagram. And myelin sheath is responsible for white color of the fibers. So nerve fiber, when we'll dissect the brain, okay, at the later stage, as I mentioned, we have to dissect the brain. तो उसमें क्या होता है, ये जो skull का part है, okay, we have to cut the skull and then we have to take out the brain. Okay, last time also it was done. Okay, and you can see it also. So with the help of sometimes it depends, with the help of chisel and hammer. Do you know what is chisel? Have you seen carpenter? Yes. So you have seen carpenter with the help of hammer. They used to give different types of shape. तो ये जो हाथ में लेके carpenter से मारता है, this is called as chisel. ठीक है? So with the help of the chisel, the skull cap is removed. Okay, but we have to take very care, or else the brain can get damaged. Okay, so after that, once the skull cap is removed, then we have to cut from the few centimeter below the medulla oblongata, so that the spinal cord can get separated, and then we can take out the brain. Okay, so Now when you will take out the brain, and after that different sections of the brain are taken to show you the different parts of the brain. Okay, so there is a important topic called this one white matter of the brain, where different types of fibers are present. So at that time you can easily notice that the neurons where it is present, it is slightly gray in color, but जहाँ पे neurons नहीं रहता है, completely white होता है. Why? Because of the presence of the fibers. Okay, and why those fibers are completely white? Because of the presence of myelin sheath. Next, the formation. Now again, this is a topic of physiology. Okay, so जो myelin sheath होता है ना, initially myelin sheath is not present. Okay, gradually the myelin sheath is formed around the axon, and that is called as myelogenesis. Again, this thing we will study in physiology. Now, in peripheral nerve, myelogenesis starts at 4th month of intrauterine life, and it is completed in the 2nd year after birth. So, complete myelination is taking place after birth. 2nd year means 2 years of birth. Okay. Now, what is the function of this myelin sheath? Now, you know that myelin sheath is present. You know how the myelin sheath is formed in case of central nervous system and in peripheral nervous system. Okay. Now, if you see the composition of myelin sheath, it is made up of lipid 80%, then protein and water. These things are present around the myelin sheath. Okay. Now, the question is, why this myelin sheath is present? What is the function? So, see, these are four important functions. First of all, protection and physical support to the axon. Though it is a covering, so it will give protection. Okay. Next, insulation of the axon, so that the impulse can pass easily, efficiently, and then increase nerve conduction by saltatory conduction. Have you heard about this saltatory conduction? क्या होता है saltatory conduction में? It is a type of nerve conduction जहाँ पे nodes of reindeer रहता है, myelin sheath है, then small part myelin sheath is absent, then again myelin sheath is present. तो जो nodes of reindeer रहता है वहाँ पे क्या होता है, the nerve impulse is to jump. Okay, so that type of conduction is called as saltatory conduction. Again, this physiology topic you will study in physiology. Next, a neurolimel sheath plays a major role in the nerve regeneration. So if there is any damage, it is also helping in regeneration of the nerve fiber. So these are some important functions of myelin sheath. Okay, so along with the myelin sheath, that is all about the structure of neuron. Okay, next we have to see the classification of neuron, because in the beginning I have told you that approximately 1 trillion neurons are present in our body. Okay, that means some classification will be there, right? So now we have to see the classification of the neuron. Now there are classification of the neurons based on different categories. For example, first is the classification done based on polarity. Now what is the meaning of polarity? Number of processes. Okay. Next, some other classifications are done. Can you tell me what are the other classification? Next will be based on the length of the axon, Golgi type 1, Golgi type 2. Next will be based on function, whether it is sensory neuron or whether it is motor neuron. Okay? So like this, different classifications are done. So here what you have to know the function, means when it is done, what is the structure of that particular neuron, and examples. Same like the epithelial tissue. In epithelial tissue, how you have studied simple, what is the structure, and what is the location or the example. The same thing will be repeated over here. So according to the polarity, first, or also called as number of process, let me show you the diagram. Here you can see, this is the classification shown based on the polarity. Okay. So polarity means number of processes. So first is the pseudo-unipolar neuron. Do you remember about pseudo-stratified epithelium? See, again, that's why I mentioned it is just like epithelial tissue. So here again you are getting the pseudo-unipolar. तो इसका मतलब क्या है? हाँ, तो see, polarity means according to the number of processes. Okay. Now in case of pseudo-unipolar, what happens? Actually one process is there which is coming out from the cell body, and after that at a short course, after having a short course, it is dividing in T junction. Okay, so here you can see this is the pseudo unipolar, this is the cell body. Now from the cell body, one process is coming out. After a short course, it is dividing into two processes. Okay, one is the peripheral process, another is the central process. Now when you will see at that time, you will notice that two processes are there, but actually these are not two processes. This is the one process, and this one process is divided into two processes. That's why appearance is bipolar, but actually is unipolar. दोनों के बीच में है, that's why it is called as pseudo unipolar. Okay, so it is pseudo unipolar, means one, not bipolar, but it appears as bipolar. ठीक है? Now what will be the example? The best example is dorsal root ganglia. I am coming, what is this ganglia? Okay, of spinal nerve, sensory ganglia of some cranial nerves. These are the examples of pseudo unipolar neurons. Okay. Next is the bipolar neurons. So can you see the bipolar neurons? See, two processes are there. So this is the cell body. Two processes are coming out. Now this is called as bipolar. Now here two processes, one dendrite and one axon from the cell body is coming out. Such neurons are found in olfactory epithelium. This is the example. Then in case of retina of the eyeball. So these are some examples of bipolar neurons. Next, multipolar neurons. This is the diagram showing the multipolar neurons. The multipolar neurons में क्या होता है? Multipolar cell body which emerges from several dendrites and single axon. So several dendrites will be there and there will be single axon. So here you can see a single axon is there and several dendrites are coming out. So this type of arrangement is called as a multipolar neuron. Majority of the neurons in our body are multipolar neurons. Okay, majority. Next, according to the length of the axon and dendrites, Golgi type 1 and Golgi type 2. Golgi type 1 में क्या होता है? The length of the axons are longer. Approximately it reaches up to 1 meter long. Okay, that is the Golgi type 1. Now axons of these neurons form the long fiber tracts of the brain and spinal cord. तो brain spinal cord में जो tracts होता है, tracts में जो neuron का fibers होता है, those are the examples of Golgi type 1. Okay. Next, you can see pyramidal cells of the cerebral cortex, Purkinje cells of the cerebellum. Okay, these are also examples of Golgi type 1 neurons. क्योंकि इससे जो fibers निकलता है, the length is more and sometimes it reaches up to 1 meter. Next is the Golgi type 2 or microneurons where axons are morphologically similar to that of the dendrites, and because of that a star-shaped appearance can be seen. That means the length of the axon is very less in case of Golgi type 2. The best examples are they found large number in cerebral cortex, cerebellar cortex and in the retina. Okay. Next is the functional classification. Now in functional classification again it is of two types, that is sensory neurons and motor neuron. Okay. Now what is the definition of sensory neurons? They carry impulses from receptor organs to the central nervous system. So I have given an example. Suppose you are suddenly you have came in contact with any hot object. At that time you will remove your hand. Now those type of sensation will be carried by sensory nerve fibers. तो उसमें क्या होता है? The impulses from the receptor are going to the central nervous system. Now it may be brain or it may be spinal cord. That is the sensory neurons. Next types of sensory neurons, again two types are there: primary sensory neurons and secondary sensory neurons. Okay. Now what is this primary sensory neurons? Now here what happens? Cell bodies are situated outside the central nervous system except in case of fifth cranial nerve. Okay. Next, secondary sensory neurons, cell bodies or the neurons lie in the central nervous system, for example brain and in the spinal cord. That is the difference of the primary and secondary sensory neurons. Some tertiary neurons are also there, where cell bodies of this neuron lie within the thalamus of the brain. So in some book it is not given separately, because thalamus is a part of brain only, but in some books it is written separately. That's why I have mentioned this tertiary sensory neurons. Okay, only the cell body is present in thalamus. Okay, now please keep that in mind. Any type of sensation in our brain, the cerebral cortex which is present, in that different areas are there, functional areas. Whatever activities you are doing, whatever functions you are doing, that is controlled by the cerebral cortex only. Now different sensation from the receptor, ultimately through the tracts, they are reaching the cerebral cortex. Sensory cortex is also there, motor cortex is also there. Now when they will reach the cortex, at that time it has to pass the thalamus. Okay, any type of sensation, once if it has to reach the cerebral cortex, especially the sensory cortex, it has to pass through the thalamus. Okay, so that's why thalamus jo hai, that becomes the tertiary one. So primary sensory will lie the outside the central nervous system, secondary will be in the central nervous, especially in the brain and spinal cord, and tertiary they are lying in the thalamus. Okay, next is the motor neurons. Now motor neurons, what it will do, just opposite of the sensory neuron. That means they will transmit impulses from central nervous system to the muscles or glands or any other organs. Okay, that is the motor neurons. Now these cell bodies of the neurons lie within the central nervous system, except those of the postganglionic neurons of the autonomic nervous system. Now in the classification I have shown you the autonomic nervous system. Okay, where from autonomic nervous system we are getting sympathetic and parasympathetic nervous system. Okay, so in case of motor neurons, the cell bodies of these neurons lie within the central nervous system mostly, except the postganglionic neurons of the autonomic nervous system. Next, in the motor neurons, again it is of two types, upper motor neuron and lower motor neuron. Okay, now in case of upper motor neuron, the cell bodies are located within the cerebral hemisphere. For example, motor area of the cerebral cortex. From there, the fibers which are coming, they will be forming the tracts. Okay, and then they will go to the spinal cord, and from there the nerves which are coming out, it will control the muscular activity, the contraction of the muscles and all. Okay, so that is the upper motor neuron. Next, the lower motor neurons are there, where cell bodies are located within the brainstem and spinal cord. Okay, brainstem, why the brainstem is mentioned, because the motor nucleus of the spine, this one, cranial nerves are situated in the brainstem. Okay, now I don't know whether you have studied this or not. ये जो face में जो muscles है, facial muscles, where different expressions can be given. When you are happy, expression is different. When you are sad, expressions are different. ठीक है? तो ये जो facial expression है, that is not, तो facial expressions के लिए क्या होगा? जो face का muscles है, उसका contraction relaxation होगा. Now that is not controlled by spinal cord. It is controlled by the cranial nerve, facial nerve. Okay, and that is coming from the brainstem. So that's why I have mentioned this brainstem and spinal cord. Okay, so for brainstem I have given one example. Spinal cord means जो limbs, I'll take the example. ठीक है? Now these things are important when you will study the neuroanatomy. At that time upper motor neuron lesion, lower motor neuron lesion होता है. ठीक है? At that time the symptoms occurring are different. Okay, next, in autonomic nervous system, if you see the motor neurons, again they are divided into two types, preganglionic and postganglionic. Now what is this preganglionic? Here cell bodies will be situated in the brain and spinal cord, and here it will be situated outside the central nervous system. For example, collateral and terminal autonomic ganglia. Now I hope you have noticed that several times I have mentioned this terminology, ganglia. Okay, now we have to study. Today we'll be discussing about the ganglia also. What is this ganglia? What is the structure of the ganglia, and what are the different types of ganglia? types of neurons are done. Next is the neuroglial cells. Here is the diagram. Here is the diagram. You can see the diagram of the neuroglial cells. Okay. So these neuroglial cells, in the beginning I have told you, these are nothing but the supporting cells, and number of neuroglial cells are more as compared to the neurons in the central nervous system. Okay. Now these neuroglial cells, different neuroglial cells are there. Among them, majority are the astrocytes. Okay. So you can see different neuroglial cells are there. So these are the astrocytes. Here you can see oligodendrocytes. You can see these are the satellite cells. You can see Schwann cells. You can see ependymal cells. Okay. Then microglia are there, or also called as glial cells. Like this, different types of neuroglial cells are present. Now each and every type of neuroglial cells are having important functions. Okay. Now if you see the astrocytes, so they are having a star-like appearance. Okay. And central body is there, and you can see the foot processes which are present around any blood vessels which are present in the central nervous system. Now these astrocytes are having very important function, that is they helps in the formation of blood-brain barrier. Okay. Now because of this blood-brain barrier only, this toxic substance whatever is present in the blood is unable to go in the central nervous system. ठीक है. जो blood-brain barrier है, that is a barrier between the blood and the central nervous system. So whatever we are eating after metabolism, it goes into the blood. You know that, right? So after that, through blood circulation, it can reach to the central nervous system, into the nervous tissue. But all the substances are not allowed to overcome the blood-brain barrier. Only few substances are permissible to overcome the blood-brain barrier. Okay? Why? To protect the delicate structure of nervous system. Okay? And in that, the major role is played by the astrocytes. So what I will do, I will request everyone to study the other types of neuroglial cells. Okay, so many things are there, it will take time to complete everything. Nothing much, only the type of the cell, its structure, what is the appearance and the function. That's it, nothing else in the neuroglial cell. Okay. In any of the histology book, if you open the nervous tissue chapter, you will get the neuroglial cells also. So I have given one example, that is the astrocytes, most abundantly present neuroglial cells in our body, and I have given the function also. Remaining cells are there. I will request you guys to study from your own. After that, if any difficulty, you can ask. Okay, so ependymal cells are also there, oligodendrocytes, microglia. Okay, so these are the different types of neuroglial cells. Next is the structure of peripheral nerve. Now this is very important. Okay, structure of peripheral nerve. Mostly in histology, if the question comes from nervous tissue, this is one of the important topic from where the question used to come. Structure of peripheral nerve. Okay. Now, if you see the peripheral nerve, it is the collection of many nerve fibers which are held together by connective tissue. If you remember the muscle fibers, skeletal muscle fibers, you will see some similarities present over here. Okay. Now, in case of nerve fiber, two types of fibers are there: motor fibers and sensory fibers. Motor fibers are also called as efferent fibers. Sensory fibers are also called as afferent fibers. In case of motor fibers, the axon carry impulses from central nervous system to the muscle or gland. Okay? That is the efferent. And from the effector organ, if it is carried in the opposite direction towards the central nervous system, then it is the sensory fibers or afferent. Okay. Now here you can see the diagram. Okay. So in the basic structure of peripheral nerve fiber, first of all, we are having each nerve fiber is having a central core which is formed by axon, and it is called as axis cylinder. central core में क्या रहता है, axon रहता है, that is also called as axis cylinder, and that is covered by a plasma membrane that is called as exolemma. Already we have discussed. Next, it is covered surrounded by myelin sheath, which you can see over here. And the sheath is in the form of short segment, and that is called, and the intervals are called as nodes of Reinbeer. And the part of nerve fiber between two consecutive nodes are called as internode. Okay. Now outside the sheath, there is a thin layer of Schwann cell and cytoplasm that can also be seen. Here you can see the Schwann cell. Now the layer of cytoplasm is called as neurolemma. That is the basic structure of nerve, peripheral nerve fiber. Now ये जो peripheral nerve fiber है, suppose you have taken one peripheral nerve, ठीक है, at that time you will come to see that connective tissue coverings are also present. Okay. Now this is the diagram showing the histology of peripheral nerve. So what you can see, so here you, first of all you can see each nerve fiber, axons are there, Schwann cells and basal lamina where the axon cells are resting. Then 3 layers of connective tissue coverings are there. Okay, it is the endoneurium, perineurium and epineurium. Now in case of endoneurium, it surrounds the individual nerve fiber. Okay, तो यहाँ पे जो individual nerve fibers, see these are the cut sections of the nerve fibers. Okay, now individual nerve fibers are, here you can see individual nerve fibers are covered by connective tissue covering that is called endoneurium. Okay, now a fasciculus or a bundle of nerve fibers again covered by connective tissue covering that is called perineurium, an entire nerve again covered by connective tissue covering that is called as epineurium. So each nerve is surrounded by a layer of connective tissue that is called endoneurium. Each nerve, one single nerve. Now the endoneurium, what it will do, it will hold the nerve fibers together and facilitate the formation of fasciculus. Now if you see the endoneurium, it is made up of fibroblast, collagen fibers, endothelial cells, macrophages, etc. These are present. Next, each fasciculus is surrounded by a thick layer of connective tissue that is called perineurium, as you can see in the diagram. Perineurium, thick layer. Okay. Next, the fasciculi are held together by a dense layer of connective tissue. Now, all the fasciculi, they are held together by a dense layer of connective tissue that is called as epineurium. Okay, as you can see, this is called as epineurium. So, if you take a nerve fiber, okay, and then if you take the TS, then such kind of appearance will be there. That means if you take one peripheral nerve, not one nerve fiber, if you take one peripheral nerve, okay, and then you have taken the TS, you have prepared the slide, H&E slide. If you see under microscope, ऐसे दिखाई देगा. Now, last time, do you remember when we have discussed about the connective tissue covering of muscle fiber, skeletal muscle fiber? So why this connective tissue covering is required? क्यों चाहिए? Connective tissue covering, why? In case of nerve fiber, we have studied that so that nerve supply, blood supply, lymphatic drainage can be done. Now here it doesn't require nerve supply, but blood vessels are coming. ठीक है? ये देखो, can you see the blood vessels? See, blood vessels are coming. Nerve fibers also require blood vessel. Without blood vessel, it will degenerate. It will start degenerating. Continuous blood supply is required. So how those blood vessels will be carried? And those are carried by this connective tissue covering. So that each and every single nerve fiber is getting the blood supply. Yeah. Now, see, this is a high magnification image of a neuron. High magnification image of a neuron. Now, what you can see over here? तो फर्स्ट यू कैन सी द सेल बॉडी. ये देखो, ये सेल बॉडी दिखाई दे रहा है, ठीक है. यू कैन सी द न्यूक्लियस आल्सो, ऑलमोस्ट सेंट्रली प्लेस्ड न्यूक्लियस. You can see the axon hillock. You can see the axon which is going like this. Okay. After that, you can see the dendrites. Okay. And along with that, around this cell body, can you see these small dots? These are nothing but the neuroglial cells. ये देखो, these are the neuroglial cells. तो whenever you will get a slide of this one nerve cell, उसमें लिखा रहेगा nerve cell. At that time, what you have to do, you have to see this particular structure. Okay. So under low magnification, it is very difficult to identify. At least you have to zoom for under 45 or 40x to see such kind of structures. Okay. और ऊपर से भी न्यूरोग्लाया ठीक से दिखाई नहीं देता है, but still you can at least identify that some neuroglial cells are also present. So why I am showing you this slide? The main aim is to know what is the appearance, because previously the diagrams which I have shown, those are the schematic diagrams. ठीक है, just see the difference. जो पहले डायग्राम दिखा है schematic diagram, अभी का जो original diagram, कितना difference है. ठीक है. Okay, now lastly, several times I have mentioned about the ganglia. Now we'll see what is ganglia. Okay, so ganglion is the aggregation of cell bodies of neurons that also outside the central nervous system. Okay, what is ganglion? It is the aggregation of cell bodies of the neurons outside the central nervous system. It is called as ganglion. Now, two types of ganglion are there: sensory ganglion and autonomic ganglion. Now, please do one thing. If you have written this line, ganglion is the aggregation of cell bodies outside the central nervous system. Okay. Now, repeat the same thing and write. If it is inside the central nervous system, then it is called as nuclei. Repeat the same line, and instead of outside, write inside the central nervous system, then it is called as nuclei. Not the nucleus which you see in the cell. That nuclei is different. Okay? समझ में आया कि नहीं? समझ में आया क्या लिखना है? हाँ. Now the question is why it is important. So as I have mentioned, when you will study the brainstem, ठीक है? When we study the brainstem का तीन part होता है: medulla, pons and midbrain. So first we see the external features of brainstem. Then we study the internal feature. In internal feature, what we do, we take the sections at different level. When you take the section at different level, at that time you will come to know that two things can be seen. One is gray matter and another one is white matter. Gray matter में nuclei दिखाई देता है, mainly different nuclei are there. At that time, that point will be there. So what is nuclei? This is nothing but the collection of cell bodies, okay, inside the central nervous system. That is the nuclei. Next, ganglia are of two types: sensory ganglia and autonomic ganglia. Okay. Now you have to know the difference. Now this question is important. Kindly mark the topic. यहाँ से क्वेश्चन आता है that sensory ganglia, what is ganglia, what are the different types of ganglia, or write the differences between sensory ganglia and autonomic ganglia. ठीक है, ऐसा क्वेश्चन आता है. Now in case of sensory ganglia, they are present outside the central nervous system. Neurons of the sensory ganglia, they carry impulses towards the central nervous system. Okay, that means from the periphery, the impulses are carried towards the central nervous system. That is the function of the sensory ganglia. Now, if you see the examples, dorsal root ganglia, or also called as DRG, of spinal nerve, sensory ganglia of various cranial nerve, for example, trigeminal, facial, vestibulocochlear, glossopharyngeal, and vagus nerve. Now, these are the cranial nerves where you will be getting the sensory ganglia. Okay, but in general, in the spinal cord, we are getting the dorsal root ganglia. Now, what is the structure? Now, first of all, जो ganglia है, तो what I have mentioned is a collection of cell bodies outside the central nervous system. Now, which type of neurons are present? In case of sensory ganglia, pseudo-unipolar neurons are present. Now, this is the most important thing. Okay, so in sensory ganglia is having pseudo-unipolar neurons. But an exception is there, that is the vestibulocochlear ganglia, where we are getting the bipolar neurons. Okay. Next, if you see the pseudo-unipolar neurons, have a peripheral process carries information from the sensory receptor, and central process that will pass the received information to the central nervous system. So in the beginning I have told you that one process, it will come and it will divide to form two processes, that is one central process and one peripheral process in case of pseudo unipolar neuron. Next, if you take a section through sensory ganglia, then you will see large neurons present in groups. So large neurons will be there and they will be present in groups. Next, if you see the nuclei, they are large vesicular and centrally placed nucleus. Okay, group of neurons are separated by bundles of myelinated nerve fibers. These are some important identifying features of the sensory ganglia. Next, satellite cells or capsular cells. These are nothing but the glial cells, neuroglial cells. They are present and they are providing nutrition, okay, to those neurons which are present over there. Next, satellite cells are flattened or low cuboidal cells, and the ganglions are covered completely by connective tissue capsule. These are some important features of the sensory ganglia. Here you can see the diagram. This is low magnification image. This is high magnification image. Okay. Now here what you can see, please concentrate over here. So this is high magnification. In the high magnification, can you see these are nothing but the pseudo unipolar neurons. These are the pseudo unipolar neurons. Okay. Now in pseudo unipolar neurons are present inside the sensory ganglia. Exception is vestibulocochlear where bipolar neurons are present. Next, mostly you will be getting centrally placed large vesicular nucleus. Next, if you see the periphery, ये जो periphery है, then what you will see the satellite cells. ये जो cells दिखाई दे रहा है, these are the satellite cells. These are the satellite cells. Now these satellite cells are completely covering the neurons. Okay, they are forming a capsule around the neurons. Now इसका function क्या है? It will provide nutrition to these particular neurons. That's why these satellite cells or capsular cells are present. Okay, and then some Schwann cells are also present. Why? Because the nerve fibers which are present will separate the group of the cells. यहाँ पे जो cells रहता है, they are in the form of groups. Those groups are separated from each other by myelinated nerve fibers. Because of the myelinated nerve fibers, you can see the Schwann cells. Okay, and complete ganglia is covered by connective tissue capsule. Complete capsule can be seen. So these are some important features, identifying features of the sensory ganglia. Okay, next is the autonomic ganglia. Let me show you the diagram. Here you can see the diagram of the autonomic ganglia. So what you can see in the autonomic ganglia. Now here also aggregations of the cell bodies are present. Okay. Now autonomic ganglia are subdivided again into sympathetic and parasympathetic. Now here the cell bodies are multipolar neurons. Okay, here the cell bodies are multipolar neurons. Okay. Next, if you see the nucleus, it is eccentrically present, not at the center, eccentrically present. Okay. Next, the neurons are separated from each other by bundles of nerve fibers, but here unmyelinated nerve fibers are present. So first of all, multipolar neurons are there. Okay. Then the nucleus which is present, that is eccentric. And if you see the size of the neurons, okay, they are smaller as compared to the sensory one. Next, the number of satellite cells or the capsular cells are less, and complete capsule is not formed. Next, these postganglionic autonomic neurons, they synthesize catecholamines and acyl granules. Okay, and they are prominently seen in the cytoplasm. Okay. Now this is the low magnification image and this is the high magnification image. Okay, and one important thing, in some cases you can see the lipofuscin granules. ये देखो, lipofuscin granules can be seen. ये देखो यहाँ पे दिखाई दे रहा है inside the neurons. ठीक है. So what you have to see, first of all, these are multipolar neurons. These are multipolar neurons. Now this is the nucleus. Nucleus is eccentric in nature. Okay. Next, if you see the cells, they are never in the form of groups. Previously we have seen those cells are in the form of groups. Next, ये जो satellite cells रहता है, okay, the capsular cells, Okay, these are incomplete or the number of cells are less in nature. Next, another important thing, can you see these granules, these dots? These are called as lipofuscin granules. Okay, lipofuscin granules. This is a special type of granules present. See, lipofuscin pigment or lipofuscin granules. तो ये क्या होता है, whenever cells are getting older, okay, तो जो cellular metabolism होता है, after cellular metabolism the debris are stored, okay, and then when the staining is done, at that time a special stain is used, so yellow brown appearance is there. Okay, that's why these are called as lipofuscin pigment. So those can be seen in some cells, not in all the cells. And if it is seen in the cell, that means those cells are older as compared to the newer cells. Okay. Now for your reference, I have added this one. This is the difference showing the, this is a chart showing the difference between the autonomic ganglia and sensory ganglia. So what you can see, in case of sensory ganglia, here the cell bodies are larger, here cell bodies are smaller. Okay? Now here pseudo unipolar and here multipolar neuron are present. Okay? Here you can see the nucleus centrally placed, here eccentrically placed nucleus. Now cell bodies of the neurons are arranged in groups, doesn't form definite groups. Here myelinated nerve fibers are present and those nerve fibers are separating the groups of the cells. Here unmyelinated nerve fibers are scattered. Then prominent satellite cells, here satellite cells are less prominent, means less number of satellite cells. A well-defined connective tissue capsule, poorly defined or absent connective tissue capsule. Okay? So this will help you to understand about the difference between the sensory ganglia and autonomic ganglia. Okay? Again, I am mentioning one thing, autonomic ganglia is of two types. What are they? Sympathetic and parasympathetic. So that is all about the nervous tissue. Now in nervous tissue, what you have to write exactly? Now in please note it down what you have to write exactly in the nervous tissue. First of all, you have to write the introductory part of the nervous tissue. Okay, first you have to write the introductory part of the nervous tissue, then you have to write the structure of neuron. Okay, along with that the diagram. And in the introduction, don't forget to mention the classification of nervous system. In the introduction, you have to write the classification of nervous system. Okay? So, next, just you have to draw the diagram of neuroglial cells. Okay? And write one function each. Means you don't have to explain everything. In neuroglial cell, just draw the diagrams of the neuroglial cell. Okay, this you don't have to draw by H&E pencil, normal pencil or color pencil, whatever you want. Okay, and you have to write one function each. Okay, तो ये हो गया neuroglial cells का. Next, come to the peripheral nerve. Here you have to write everything about the structure of peripheral nerve, whatever we have discussed, along with the diagram. Now here you have to use H&E pencil. Okay, so which diagram you have to draw in peripheral nerve? This one, this diagram. Okay, in peripheral nerve you have to write histology of peripheral nerve and this both diagram. ठीक है, दोनों diagram draw करना है. And the structure, entire structure. Now whenever you are writing the structure, make sure that you are mentioning about endoneurium, perineurium and epineurium. ठीक है? Next, about the ganglia. For ganglion, you have to start from here. What is ganglia? Definition of the ganglia. Then types of ganglia. Here sensory explanation. Then diagram. Now, please listen one thing. Here two diagrams are given. ठीक है? So in low magnification, it is difficult to identify. That's why you can draw this diagram under high magnification. But it should be HNE diagram. Okay, only get this one, high magnification. Okay, next autonomic ganglia also. This part, this here you can draw both the diagrams. Okay, because here also things are visible, there also things are visible. You can draw both the diagrams. And lastly, you have to write this chart. Okay, showing the differences between sensory and autonomic ganglia. So that is all about the nervous tissue.